Have you ever really thought about how a doctor can find one single tiny mistake in your DNA? I mean, a mistake that could literally change everything about your life. It's got to be one of the most amazing detective stories in all of modern medicine.
Okay, so here's the real challenge we're up against. Your genetic code, your entire genome, it's like a story that's 3 billion letters long. So how on earth do you find a single life-changing typo in all of that? You know, we're not just looking for a needle in a haystack here. No, it's more like trying to find one specific blade of grass in a haystack the size of a mountain.
And that number right there, that is the mountain. 3 billion. That's the sheer, mind-boggling scale we're dealing with. To find just one error in this ocean of information requires this incredible mix of technology and, well, just absolute precision. So where do we even begin with a problem this big? Well, the whole journey starts with the raw data, the stuff that comes straight off a DNA sequencing machine. The very first thing we get back from the lab is this file called a FASTQ file. Now, the best way to wrap your head around this is to picture that 3 billion letter book of your life, and then imagine feeding it into a paper shredder. Seriously, the DNA sequencer can't read the whole thing at once. It reads it in billions of tiny, shredded, overlapping pieces. This FASTQ file, it's basically that giant pile of digital confetti, but with a little bonus. Each piece comes with a quality score telling us how sure we are that we read each letter correctly. All right, so now we have this massive digital pile of shredded paper. The next phase is absolutely critical. We have to meticulously tape that entire book back together in the right order. And this isn't just one magic step, you know? It's a highly structured data assembly line. First, we have to check the quality of all our shredded pieces. Then we align them. After that, we get rid of any accidental photocopies. And finally, we kind of double-check our confidence in the whole assembled text. Let's actually zoom in on a couple of the most important steps here.
This first quality check is completely non-negotiable. See, a Q30 score means there's a 99.9% chance that a letter was read correctly. For any kind of clinical analysis, we demand that at least 80% of all the letters in our raw data meet this really high standard. If they don't, it's garbage in, garbage out, the whole thing is a wash.
This is how we solve that shredded book problem. Once we know our data is good enough, we start the alignment. We take a standard reference human genome, think of it as our master copy, and we map every single one of our tiny fragments to its correct spot, piecing the whole story back together, bit by bit.
Okay, the book is reassembled. Now we can finally get to the most exciting part, hunting for those typos. These are the genetic variants that make you, and sometimes can be linked to disease.
And there it is, right there. This is what we've been looking for the whole time. On the top, you've got the reference sequence, and on the bottom, the patients. See it? That T has been swapped out for a C. That single letter change is what we call a single nucleotide variant, or an SNV for short. So the big point is, we found a difference. But is it real?
Well, to make that call, scientists use some unbelievably sophisticated software. The gold standard in a lot of labs is something called the GATK HaplotypeCaller. It's really clever. It doesn't just look for simple differences. It actually weighs all the statistical evidence for that variant across all those tiny overlapping fragments that cover that one specific spot in the genome.
But, just finding a potential variant, that's only the beginning. In a clinical setting, that finding has to survive a grueling gauntlet of quality control checks. We have to be sure it's a real biological signal and not just some random digital noise. And this isn't guesswork. It's a checklist with hard numbers. Take mean coverage, for instance. That tells us how many times, on average, each letter in your DNA was read. For a clinical test, we need to see it at least 80 times to be confident. And the mapping rate shows us that at least 95% of our shredded pieces found their proper home. Every single analysis has to pass these strict thresholds, no exceptions.
So how do we know this whole assembly line even works? We validate it. We constantly run samples that have known typos through the entire system, and we demand that it finds at least 99% of them. That's what we call sensitivity. We also demand that at least 99% of the typos it calls are actually real, and that's precision. The standards here are just incredibly high.
All of these steps, right from that first raw data check to the final variant validation, they're all designed to do one single thing: build a result that a doctor and a patient can trust with their lives. And that trust? It's built on traceability. Every single report comes with this detailed audit trail. We know exactly which version of the software was used,
who ran the analysis, and on what day. This makes the entire process totally transparent and reproducible, which is absolutely essential for clinical accountability.
You know, part of building trust is also being really honest about what you can't see. The final report will always explicitly state the limitations of the test. So this specific method is amazing at finding these small typos, but it's not really designed to find huge, large-scale structural changes or variations in very complex, repetitive parts of the DNA. Transparency is everything. So in the end, finding that one typo in 3 billion letters isn't about some magic button. It's about a rigorous, validated, and transparent process where every single step builds on the last. It creates a final report that isn't just data, it's a foundation of trust. And that really leaves us with a final thought, doesn't it? As our ability to read this book gets better and better, how are we going to choose to edit the story?